Hola! Today we are going to learn about insecticide resistance, mechanisms, and management. So let's start. Resistance overview, evolution of resistance, factors promoting the development of insecticide resistance, resistance mechanisms, IRM, that is insecticide resistance management, BT crop resistance management. Resistance overview history 1908 San Jose scale to lime sulfur in Washington, DDT resistant houseflies in Sweden 1946, 1997 over 500 arthropod species resistant to one or more insecticide groups. 1997 over 500 arthropod species resistant to one or more insecticide groups. So resistance is kind of increasing. Okay, resistant species are also increasing. What is it? The development of an ability in a strain of insects to tolerate dosage of toxicants which would prove lethal to the majority of individuals in a normal population of the same species. It's about a pest population, not an individual. Reduction in the susceptibility of a population to pesticides. It is genetically based. Heritable reduction in susceptibility. It is not susceptibility difference known as tolerance between different stages of feeding on different plants. Resistance overview. The champions: diamondback moth, Colorado potato beetle, green peach aphid, whitefly, spider mite, cotton bollworm, mosquito. Resistance overview, type of resistance, multiple resistance. Resistance to multiple insecticides of different classes by multiple mechanisms, consequences of sequential application of insecticides, pesticide treatment. Cross resistance, resistance to one insecticide leads to resistance to another yet unused insecticide. Usually the two insecticides belong to the same class and share identical or similar mode of action. Negative cross resistance, resistance to insecticide A leads to susceptibility to B and vice versa. Evolution of insecticide resistance, how does it occur? Individuals in a population are never equally susceptible to an insecticide. Although initially rare, insecticide resistance R genotype is present. Frequency is 1 by 12, that means 0.083. Okay. Evolution of insecticide resistance. An insecticide is used, leaving insecticide resistant individual their R genotype and some susceptible individuals S genotype. Now the frequency of resistance R genotype is 1 by 3. That means 0.333. Evolution of insecticide resistance. If R genotype reproduce as well as S genotype, in the next generation, the frequency of R will be the same as the survivors in the preceding generation. That is 0.333 by 3. Evolution of insecticide resistance, R genotype frequency, generations. Evolution of insecticide resistance, when LD50 ratio resistance occurs through insecticide selection. Selection acts on genetically based variation in susceptibility which arise from mutation. The source of all new genetic variation, genetic recombination that rearranges genetic variations. Gene flow from populations having different allelic frequencies. Persistence of resistance. Resistance takes a while to develop. Suggest R alleles are initially rare. The rarity of R allele suggests it comes from reduced fitness. Thus, R alleles should fall to a very low frequency without insecticide selection. rarity of R allele suggests it comes with a reduced fitness. This R allele should fall to a very low frequency without insecticide selection. Dieldrin resistance in Australia sheep blowfly declines due to low overwintering survival. Some resistance are fixed. DDT resistance fixed in many insects like housefly, Drosophila, pink bollworm. Dicophore, structurally similar to DDT, resistance in European red mite. Then bendicarb, a carbamate, resistance in many strains of German cockroaches. Factors influencing evolution of resistance: genetic factors, biological factors. Then operational application factors. Genetic factors influencing evolution of resistance: number of R gene, quicker if one major gene, dominance of R allele, quicker if R allele is dominant, initial frequency of R allele, the higher the quicker. Fitness of R allele, quicker if no fitness caused. Biological factors that promote resistance: short generation time, high fecundity, no or little migration occurs within populations. The species is highly mobile, increasing the possibility of exposure to insecticides. Biological factors that promote resistance: short generation time, high fecundity, no or little migration occurs between populations. The species is highly mobile, increasing the possibility of exposure to insecticides. Optional factors that promote resistance: low economic threshold, use of the same insects every generation, a large geographical area is treated, high dose, no refuge exists, long persistent insecticide or slow release formulations. Use insecticides related to one used earlier. Resistance mechanism: how do insects become resistant? Insecticide activated, detoxified, sequestrated, excreted, target sites, intoxication, death. Behavioral resistance: avoiding toxin. Detect and avoid the toxin. Anopheles mosquito lives and bites inside home, but RR remains outdoor and flies into house to bite because DDT was applied to interior walls. Houseflies avoid treated surface. Cockroaches avoid treated surfaces and baits. Diamondback moth avoid permethrin. Reduced penetration. RR individuals have a pen gene that reduces penetration of insecticides 2 to 3 fold due to modified composition, modified structure. Usually present along with metabolic and or target mechanisms to apply resistance. Insecticide sequestration: integument, epicuticle, exocuticle, endocuticle, epidermis, fat body, perivisceral fat body, peripheral fat body. Increased direct excretion. Insecticides excretion: tobacco, hornworms. Increased metabolism. The most common resistant mechanism called metabolic resistance. RR individuals have more enzymes or more efficient enzymes. P450, cytochrome P450 monoxygenase is involved in metabolism and resistance to all classes of insecticides. Esterase is involved in the resistance to organophosphate, carbamates and pyrethroids. GST involved in resistance to DDT, OP and PY. P450 esterases reduced target sensitivity, target site insensitivity. The second most common mechanism is called altered target site resistance. Acetylcholine E insensitivity, resistance to OP and carb. Sodium channel insensitivity, knockdown resistance to DDT. GABA receptor channel insensitivity, resistance to cyclodine and fipronil ivermectins. AC acetylcholine R insensitivity, resistance to nicotine, neonicotinoids and spinosad. Molecular mechanisms of resistance: Amplification is trace a mosquito's green pea japid, altered expression, structural change. Resistance mechanisms, behavioral resistance, penetration resistance, increased sequestration and direct excretion, metabolic resistance, P450, esterase, GST, altered target site resistance, ACE, ACHR, sodium channel, GABA receptor. Insecticide resistance management, IRM. What can you do about resistance? Historically, growers respond to resistance by increasing dosage and frequency of application, which only accelerate the development of resistance. Switch to another insecticide, so begins the pesticide treadmill. In theory, resistance can be minimized by increasing survivorship of SS individuals, decreased survivorship of resistant individuals. RLA. 3 IRM strategies: moderation, saturation, multiple attack. IRM by moderation strategy conserves susceptible alleles by reducing selection pressure in the following ways: higher AT, lower dosage, less frequent application. Local rather than area wide applications, preserve refuge by treating only hotspots in a field. Use short residual insecticide and avoid slow release formulation. Good for environment and natural enemies, but not practical for high value crops, fruit, vegetables, and medical insecticides. Management by saturation. Suppress resistant alleles by maximizing the mortality of R individuals in the following ways: high dosage, apply at the most vulnerable stage of pests. Age specific expression of resistance, low resistance level at higher stages. Synergies to inhibit detoxification enzymes work if R allele is recessive and rare. Environmental concern necessary for high value crops and medicinal plants. Management by multiple attack strategies suppress resistant alleles by diversifying selection forces and complicating pests adaptation in the following ways: mixed use of insecticides at low dosage that differ in mode of actions. A resistant individual can be killed by insecticide B and vice versa. The insecticide rotation, crop resistance, or multiple resistance can happen. Necessary for high crop value crop medical pests. The best solution: integrated pest management, chemical, biological, and cultural. Combination of cultural, chemical, and biological method control tactics. Diversifying selection forces and complicating adaptations. Resistance to one tactics can be compensated for by other tactics. Resistance to crop rotation, one generation per year. Adults lay eggs in the field, cornfield, diposing eggs hatch the following spring and feed on corn roots. Corn soybean rotation. Crop rotation no longer effective because northern corn rootworm extend its egg diapause from 1 year to 1 to 5 years. Western crop rootworm leave a cornfield to lay eggs in soybean or other crop fields. Repeated transgenic crops, high resistance risk. Bacillus thuringiensis toxin gene, CRY1AC, introduced into plant. Simplified toxin, 1 or 2 Bt toxins in plant versus 5 toxins produced by bacteria. Persistent selection, continual production in plant versus sprayed Bt degraded rapidly, massive plantations, high sensitivity, high resistance risk. High dosage refuge strategy. Resistance due to one gene. R allele is recessive. R allele frequency is low. R and S individuals mate randomly.